So that anti-parallel structure of the double helix affects replication. So DNA polymerases add nucleotides only to the free 3' end of the growing strand. And so whatever strand is getting built can only be built in the 5' to 3' direction. So things are going to be added at the 3 end, and that's going to be the one that grows. So you'll see that it'll only elongate in the 5' to 3' direction, meaning like it's only getting stuff added at the 3', and that's how it grows. So if we go back here, you could see that. So this template strand has to be going 3 to 5. And then the one that you're growing has to be going anti-parallel from 5 to 3. And you just add more nucleotides on the 3' end to the next 3'. And it can only go that way. And so along one template strand of DNA, so again, the parental strand is called the template strand, then the DNA polymerase is building the new strand continuously on one side. And so this side is called the leading strand. And you'll see what I mean in a second. So right now, the leading strand is kind of what we're thinking about. You're building a strand and it's just getting built longer and longer. And so that strand is called the leading strand. But then on the other half, it's different. So I'll show, in the next couple slides. So if you notice, here's the replication bubble. This strand is 5 to 3. And so the strand that you're building is the opposite direction. So this is the 5' end and it's growing the 3' end, right? And so that's a pretty clean build. You just keep going. You put down a primer and you just keep going and you keep unzipping, and then you can keep growing the strand longer and longer. So that's why it's the leading strand, because it's going ahead. But then you can see on the other half below, because it can only be grown 5 to 3, you go like this. But then there needs to be primer. So then you have to like start and stop over and over. And so that other strand is called the lagging strand because it takes a little bit to like make each part and then link them together. It's kind of like lagging because it's like that. So that's what I mean. So the template strand is the parent strand that you're reading and it's being built off of. So that's a template. And so one of them is the leading strand, the other one is going to be like a lot of starts and stops, and that's going to be the lagging strand. So they're building in opposite directions because they have to be built, 5 to 3. So to elongate the other new strand, the lagging strand, DNA polymerase has to work in a direction away from the replication fork. So it's kind of moving in the opposite direction instead of moving in the same direction at the fork. So the lagging strand is built in a series of segments called Okazaki fragments, named after the guy who discovered them. And so they're built into little fragments and then they're joined together by an enzyme called DNA ligase. So it kind of like puts it all together. So here is how you're building the lagging strand. So the leading strand gets to be built towards the replication fork, and so it gets to just keep growing. But this one, because you're kind of like building 5 to 3, it kind of has to keep starting and stopping. Why it developed like that, I don't know. That's just natural selection happens, and this is the way that things form, and it makes it complicated for us to learn about, but that's how it is. So primase has to make a primer, and it goes from 3, it builds from 5 to 3. It adds stuff at the 3' end. And then there has to be, eventually it's going to run into the leading strand of the next half. So in this direction, this half gets to go that way. So primase makes a primer, Step 2, DNA polymerase. And there are actually multiple ones, but specifically it's DNA polymerase 3, so DNA pol 3, makes this Okazaki fragment. And then DNA polymerase detaches, like it lets go because it's running into the next segment. So then it has to kind of like get up and then go backwards and then build the next part. So it goes back over here, fragment 2 has a primer put down, and then the second Okazaki fragment gets built by DNA polymerase, lets go, goes back, maybe it makes a third part over here. And then the RNA primers don't stay forever. They have to get taken off. And then, so when the primer comes away, DNA polymerase replaces the RNA that was there with DNA. So now there's DNA. And then DNA ligase joins the two fragments. So this is still true of the RNA primer needing to be taken off. It's still true of the leading strand as well. That's just not shown here. But yeah, so the Okazaki fragments and then whatever strand is next, they all get combined by removing the primers, filling in those like holes or gaps with DNA, and then DNA ligase like sticks them all together again. So this is, in summary, like a super overview of everything, all the key players and kind of the order of events. So helicase unzips the DNA. The single-strand binding proteins hold on to the DNA and stabilize the single-stranded DNAs. Primase puts down a little RNA primer, and that's why it's in red. And that helps DNA polymerase to then start building. And then DNA ligase joins all the different fragments from like the leading strand, and then the leading strand on this half to the Okazaki fragments here, and the lagging strands. And so here's a summary of all those key players. And so to point out, DNA polymerase 3 is the one that builds the new DNA strand. And it's actually, more specifically, DNA polymerase 1 that removes the RNA nucleotides of the primer and then replaces them with DNA. So there are more than one DNA polymerase. Yeah. So all these proteins that take part form a large complex called, it's like a DNA replication machine. So it makes like a whole complex. And this DNA replication machine can be stationary during the process, meaning like it's not the machinery and all the proteins that move, it's actually staying still, and it's the DNA that kind of gets pulled in and fed through and moved. So yeah, recent studies support a model in which DNA polymerase molecules are what like reel in the parental DNA and extrude newly made daughter DNA molecules. So it's not that the whole machine is moving along the whole strand. The machine's there and then it's kind of just like pulling it through. So this is what that's kind of showing. And so this is the trombone model of the DNA replication complex that we think is happening right now. Like all this forms a big complex like this and stays in place. And it has connecting proteins connecting the different parts of this complex. And then the DNA is actually what's getting like pushed through or pulled through in one way or another. And so this is another summary slide showing, again, those key players. So most of the time, this is really good. This is fairly accurate, and we'll talk about that in a second. But so most of the time it's pretty good, but there can be mistakes made. So the DNA polymerases proofread the newly made DNA and replace any incorrect nucleotides. So say there's an A, and for some reason, even though it's supposed to be a T, maybe accidentally, there's a G there or a C. So the DNA polymerases will proofread, take out any incorrect nucleotides, and then replace them. And so that is kind of like the first line of defense, like the proofreading. Next, say something happens to get past the proofreading process and not get caught. A second line of like repairing DNA would be mismatch repair. So in this case, there are repair enzymes that replace incorrectly paired nucleotides that have evaded the proofreading process. So it's like somebody else proofreading. So the DNA polymerases kind of proofread their own work. And then in the second case, in mismatch repair, it's kind of like somebody else's job, that there are repair enzymes coming through and double checking and then repairing any mismatch. And then say that there's some damage to the DNA later. So it's not like a case where there was something incorrectly put in DNA replication. If something happens to DNA at other times, it still needs to be repaired. So DNA can be damaged by exposure to harmful chemicals or physical agents like cigarette smoke and X-rays. And sometimes it can even undergo spontaneous changes. So then there's another mechanism to repair this case. So in nucleotide excision repair, a nuclease, so another enzyme that's specialized in like cutting DNA, a nuclease will cut out and replace damaged stretches of DNA. So in this case, it's not usually just like one nucleotide that's wrong, it's probably more. And so it'll cut out a whole stretch of DNA and then replace it. So again, say that this is damaged by too much UV rays. Somebody was out in the sun for too long and UV rays can do damage to DNA. And so say there's damage here at these two nucleotides. So an enzyme called a nuclease will come and cut even a little bit more outside. So even if it's technically only these two nucleotides that are damaged, nucleases will cut a little bit more. It'll cut a stretch of DNA just to be safe, cut out that whole stretch, and then you can be sure you're fixing everything. So a nuclease will cut that out, and then DNA polymerase will come in and fix it and replace it with the correct nucleotides. And then DNA ligase will again combine it and link it all together. So those are the basically three mechanisms of DNA repair. Is proofreading. So the DNA polymerases are proofreading their own work. Like if you were to write an essay and you read it out loud and you look for any errors, you're proofreading. And so you're replacing anything that is incorrect, the incorrect nucleotides. So they proofread their own work, basically. And then in mismatch repair, it's other enzymes, repair enzymes, that replace the incorrectly paired nucleotides that have evaded the proofreading process. And nucleotide excision repair is different because it's usually later on. It's not like right after any errors in replication. It's damage caused by other things like, daily life, harmful chemical or physical agents, spontaneous changes. And so they replace a little bit more than just those wrong nucleotides. And so the error rate after proofreading and repair is low, but it's not zero. So it's pretty remarkable, like how accurate it usually is, but it's also not 0% error rate. So sequence changes can become permanent and can be passed on to the next generation, especially if any of these happen in like a germ cell that's going to go through meiosis. Then that for sure, those are specific cells, the gametes, that are going to go through fertilization and give rise to a whole other offspring. So if any sequence changes happen there, then yeah, that could be passed on to the next generation. But in a way, it's kind of good. A lot of times it could be bad, but it could also be beneficial. It really is just random and depends on where the error happens. But ultimately, mutations are the source of the genetic variation that natural selection operates on. And mutations are ultimately responsible for the appearance of new species. It's just tiny, tiny, small changes that might or might not lead to a change in a phenotype. And that phenotype that's new might or might not end up being beneficial to an organism. So it really is amazing. It's so interesting how much like the molecular stuff that we're learning about in this class is what leads you to have new species and a whole phylogenetic tree and everything that you're going to learn about later on as a biology major. It all starts from DNA. It's mutations in the DNA that give the variation that ends up being what makes possible like new species and new branches, basically, because of natural selection. It's super interesting. And so for linear DNA, like again in ours, we have linear DNA molecules and chromosomes versus bacteria and prokaryotes who have circular chromosomes. So for linear DNA, the usual replication machinery can't complete the 5' ends of the daughter strands because remember, there has to be a little primer at the 5' end of a strand so that DNA polymerase can start building the new daughter strand. And so in that case, when that gets like cut off, the primer gets taken off, there is no 3' end of a pre-existing polynucleotide for DNA polymerase to add on to. Like there's nothing upstream of that. So repeated rounds of replication will end up producing shorter and shorter DNA molecules with uneven ends because it's getting cut off a little bit every time replication happens. So I'll show that in the next slide. And so, yeah, this is not a problem for prokaryotes because most of them have circular chromosomes, but it's mostly for, organisms that are eukaryotic and have linear DNA. So here is a picture showing that. So say this is the very, very end of a chromosome. And so this end of the chromosome has a 5' end. This end of this chromosome, if we were to zoom in on this lagging strand, there is a 5', there's a primer here on the 5' end. And so that's necessary to help build a little bit of the next Okazaki fragment and then the next fragment. But so when that primer gets taken off, it would normally be replaced by, DNA polymerase, but there's nothing up here to keep building off of. And you can't add stuff at the 5' end over here. You have to add stuff at the 3' end, but there's nothing before this strand. So it can't build more here. So it's just gonna be uneven like this. And then when this cell ends up duplicating this chromosome again, this time it's gonna start here and then it gets cut off. And so the new strand is going to be shorter. So eventually after so many rounds of replication, like it's going to get a tiny bit like shorter and shorter every time. Remember the primers are like 5 to 10 nucleotides. So it's not like a lot, but after so many rounds of replication, the daughter molecules are going to get just like tiny bit shorter and shorter and shorter and shorter. Yeah. So eukaryotic chromosomal DNA molecules have special nucleotide sequences at the ends called telomeres. So telomeres kind of like evolved to help prevent this shortening and shortening. It doesn't necessarily prevent it, but it prolongs the effects. So telomeres don't prevent the shortening of the DNA molecules, but they are just kind of like fluff, like random stuff that doesn't code for anything specifically. So it's fine if it gets cut off because it didn't do anything anyway. So telomeres just kind of postpone the erosion of genes near the ends of the DNA molecules. And it has been proposed that shortening of telomeres is connected to aging. So here is a little picture. They kind of look like little Among Us guys, right? And so these are the chromosomes, and the little yellow spots are the telomeres at either end. So it's really just like short sequences that don't really code for anything, actually. It's kind of just like fluff. And then if that gets cut off, it doesn't matter because it doesn't code for anything important. So if that gets cut off shorter and shorter, it's okay. It postpones the erosion of genes near the ends of the DNA molecules. And that's pretty important. It's helpful because if chromosomes, especially in germ cells, so again, germ cells are specialized cells that will end up dividing to become gametes. If those got shorter every single cell cycle, then eventually, like, essential genes would be missing from the gametes that they produce. So in germ cells, there is an enzyme called telomerase. And so that enzyme adds more telomeres in germ cells. So it's kind of adding more and more fluff so that it gets prolonged even longer. And so in normal cells, you don't have telomerase doing that, like adding more and more telomeres. It's only in germ cells. And that we think is because the shortening of telomeres might end up protecting cells from cancerous growth by limiting the number of cell divisions. It would be great to live forever, but that's not actually good. And so, yeah, we kind of think that the telomeres are shortened and shortened on purpose so that it limits the number of cell divisions. And there is evidence of telomerase activity in cancer cells, which is what might allow some cancer cells to persist. So, for example, there is a cell line called the HeLa cells, and they have an overactive telomerase, and so they can divide indefinitely in a petri dish as long as you keep feeding them and stuff, because telomerase is overactive and keeps adding telomeres, and it's not limiting the cell growth, divisions, things like that. So lastly, tying DNA back to the idea of chromosomes, the chromosomal basis of inheritance and the molecular basis of inheritance. So the bacterial chromosome is a double-stranded circular DNA molecule associated with a small amount of protein. And then meanwhile, eukaryotic chromosomes have linear DNA molecules associated with a large amount of protein. And also location-wise in prokaryotes like bacteria, the DNA is supercoiled and found in just like a region of the cell called the nucleoid, as opposed to in eukaryotes where they're going to be in the nucleus, most of it. And so in the eukaryotic cell, DNA is precisely combined with proteins into a complex called chromatin. So the DNA wraps around these proteins called histones. And so the DNA plus proteins equals chromatin. And then chromosomes fit into the nucleus through an elaborate multi-system level of packing. So here's kind of just like loose DNA in this figure wrapped around these histone proteins, and then they get wrapped even tighter and wrapped even tighter. And then that's how they get put into chromosomes. And that's how they can all fit into the nucleus. And so those proteins are called histones, and they're responsible for the main level of DNA packing in the interphase chromatin. So usually chromatin is a little bit more loose, but then when it comes to interphase, they're going to get more packed. And then unfolded chromatin resembles beads on a string, with each bead being a nucleosome. So let me show a picture on the next slide really quick. So DNA, and then histone proteins. And so when DNA is wrapped around the histone proteins, it's called chromatin. And then each little bead, or one section of DNA wrapped around histones, is called a nucleosome. So yeah, from far you can see how it kind of looks like beads on a string. And this is what it looks like under the microscope, and that's how it kind of got that characterization. And so each bead is a nucleosome, and a nucleosome is made of DNA wound twice around a core of 8 histones. So in this other picture, they look kind of flat. And there's 2 layers. There's 1, 2, 3, 4 on one half, and then on the other half, there's another 4. But here they're kind of depicted differently. So there's a total of 8. 1, 2, 3, 4 on this side, and then on the other side there's another 4. So a total of 8. And so there are 4 main histone types, and so there's 2 each, making that total of 8. And if you saw in that picture, there is like a little tail sticking out of that purple shape. And so the amino end of each histone, so the histone tail, extends outward from the nucleosome and is involved in regulation of gene expression. So here you see that these little histones, and then they have little tails coming out. And so you can modify those tails to either like make it wrap more loosely or more tightly. And that affects gene expression, because how am I going to express something if I can't even reach that gene, you know? So yeah, most chromatin is loosely packed in the nucleus during interphase and then condenses prior to mitosis. And so the loosely packed chromatin is called euchromatin. This is like a super simplified figure. This isn't even showing the 4 different histones on each side. But here you could see that euchromatin is like more loosely wrapped, and then heterochromatin is more tightly wrapped. So during interphase, a few regions of the chromatin, especially the centromeres and telomeres, are highly condensed into this form called heterochromatin. So it's still chromatin, it's just like the prefix says how tightly it's wrapped. Euchromatin more loose, heterochromatin more tight. So this like dense packing of the heterochromatin makes it difficult for the cell to express genetic information in those regions, because yeah, it's really hard to access those parts if they're wrapped by proteins really densely. And so interphase chromosomes occupy specific restricted regions in the nucleus, and then the fibers of different chromosomes don't become entangled. And then chromatin undergoes changes in packing during the cell cycle. And as the cell prepares for mitosis, the chromatin is organized into loops and coils and eventually condensing into short, thick metaphase chromosomes. So again, going back over here, here's how it looks during prophase. And then this is one loop. And then when it's coming time for metaphase, it starts wrapping more tightly. So this is all loose, but then if you wrap it like tight and then around itself, then this helps condense it a lot more and more and more until it looks like what we think of when we think of chromosomes in metaphase. And so this is showing how you can apply different like stains to paint different chromosome sets, different colors. And then this is how you would make a karyotype, is you can apply these different like things to mark where certain chromosomes are, restricted to, so they don't get tangled. And then if you were to kind of like break open this nucleus and spill out all the chromosomes and match them up, then you could see, okay, yeah, this is the set of chromosome 1. They have the same genes coding for the same traits, and then so the dyes are going to stain it this color, and this set the dye is going to stain red. Yeah. So that's kind of showing those chromosomes, and they should be packed. So these cells would be like stuck in metaphase, because that's when they're going to be tightly packed like that, right? And so those histone proteins can undergo chemical modifications that will result in changes in how condensed they are. And these changes can have multiple effects on gene expression. So we will talk a lot more about like regulation of gene expression in a few lectures, but this is kind of laying the groundwork for that. So that's everything. Hopefully now you can describe the structure of DNA, kind of roughly describe like how do we know DNA is the genetic material. Explain the Michelson-Stahl experiment and how that helped teach us about DNA replication, be able to describe DNA replication more importantly, and then distinguish among the levels of chromatin packing in a eukaryotic chromosome. So that is lecture 14, DNA.